Start recording. All right. Thank you all for joining fourth NAIRR Pilot Research Showcase. I'm Nitin Sugija. I'll be the host for this particular session. And this session is organized by NAIRR Pilot User Experience Working Group. What I mentioned, talk to you about the panelists. I would like to thank the panelists for joining us and giving up their time to give their and present their wonderful presentations. So our first guest is Dr. Andrei Irimia. He's going to talk about the project Generative Models of Neuroimages for Personalized Assessment of Risks for Alzheimer's Diseases. Dr. Andrei Irimia is an associate professor in Leonard Davis School of Gerontology at University of Southern California, with courtesy appointments in biomedical engineering and quantitative biology. His research focuses on using deep learning, generative AI, and large-scale computing to study brain imaging, traumatic brain injury, and Alzheimer's disease. Dr. Irimia leverages advanced neuroimaging and quantitative methods to understand individual variability in aging trajectories and dementia's risk. He leads several NIH-funded projects examining how chronic disease variables and women's health factors influence brain aging and neurodegeneration. Work in Dr. Irmia's lab bridges population neuroscience and clinical neurology with the goal of improving early detection and stratification of patients at risk for cognitive decline. With that, I would like to welcome Dr. Irimia and ask him to share his screen. Thank you.
Thank you very much, and thank you for having me today. It's a pleasure to be here and to speak about our research in the Leonard Davis School of Gerontology. I am a gerontologist and neuroimaging researcher, and for a number of years, I've been studying how we can use neuroimaging to estimate risk for dementia. And as you may know, there is a lot of research and interest in this specific area, given that there are a lot of protective factors that may slow down aging and risk factors that could accelerate aging. And so from the protective factors category, we can think of diet and exercise or advanced educational attainment. In the category of risk factors, you have smoking, drinking, and living a sedentary life. And so when we look at the aging process, advanced aging is associated with higher risk for neurodegenerative diseases, including dementia. And so if we can estimate the biological age of the brain, which is reflective of protective or risk factors, we can better estimate risk for dementia as well. So this is one of the motivations for this research. And one concept that helps us with this is the concept of brain age. So if we look at chronological age, at the calendar age of a person, and we look at two people who are both of chronological age 35, one of them who has high risk factors for accelerated aging might have a biological age of 40. In other words, their brain age, their neuroanatomical brain age, might be such that their neuroanatomy looks like the neuroanatomy of a 40-year-old. But a person who lives a healthy lifestyle might have a biological age of 30, reflecting slower rate of aging and perhaps low risk for dementia. So if we look at a population of individuals who are all of the same age, chronological age 38, and we look at their biological ages, we see that actually they have a normal distribution of biological ages. About half of them have biological ages younger than their chronological age, and the other half are biologically older. So in my lab, we use brain age and other approaches to better estimate risk for dementia using neuroimaging. And traditionally, we have been using MRIs to estimate brain age and risk for dementia using methods like typical regression. So in this approach, we take a look at the properties of the brain, properties of the cortex, such as cortical thickness, surface area. And in the classic approach, you can do a regression model that helps you to estimate the age of the brain from these features. More recently, with the advent of deep neural networks, we can take advantage of the power of these approaches to identify features of brain anatomy that are going to help us estimate brain age, and that might be very subtle, such that most people may not be able to identify them easily, or even experts may not be able to find them on an MRI. But because neural networks, and particularly convolutional networks and vision transformers, are able to identify these patterns easily, they can much more they can identify and estimate brain age with much greater facility. So today I'd like to show you a few of our results from this research that involve generative models for brain evolution. This is work mainly by my student Nikhil Chaudhary, and Nikhil has developed a generative AI model to forecast human brain aging and to potentially help predict the onset of cognitive impairment and dementia. And so he has looked at a large and growing sample of T1 weighted MRIs with two time points from the UK Biobank, which has over 45,000 MRIs from aging adults in the United Kingdom. And he's developed a conditional autoencoder that can take in the baseline first time point T1 weighted MRIs and conditioned on the baseline age of the individual. He can then use this the encoder decoder architecture to predict the follow-up MRI some years down the line. And the idea is that if we can estimate or reconstruct the MRI of the brain for this person later on, we can potentially estimate what their biological age will be in the future, or what their anatomy might look like, so that we can determine whether this person is at low versus high risk of dementia. So this research is very timely because if we are able to estimate the future MRI of a person, we can potentially identify those individuals who are high risk for dementia, and that can help clinicians in their strategy to improve outcomes. So here are some examples of preliminary results. On the left-hand side, the first row at the top shows the genuine MRI at follow-up of the person being scanned. And then in the middle row is the synthetic MRI. So that's the prediction of the generative AI model. And you can see at the bottom, the percent difference between two scans is quite low, such that, as shown on the right, we're able to estimate the difference in volumes between genuine and synthetic structural volumes. In some cases, we're getting very extremely low error, next to 0% for cerebral white matter. And then we have different errors looking at different portions of the brain, especially parts of the brain that have larger variability in their anatomy tend to have larger errors. And so we're working now on these models, generative AI models, that can help us to better estimate the future anatomy of the individual and help us with our goal to predict risk for Alzheimer's. Another interesting application that I'd like to show you is Nikhil's model to synthesize T2 weighted MRIs. So that's a different weighting, a different type of MRI imaging from T1 weighted MRIs, which are the standard and most commonly acquired type of volume in this field. T1 weighted MRIs are more commonly acquired, but T2 weighted MRIs can better distinguish tissue types in some cases and identify lesions, assess myelin content in the brain, which is important in aging and Alzheimer's. And so in many studies, it's very difficult or impractical to acquire too many weightings of MRIs for a person's brain. So there's a lot of interest in potentially using one type of MRI, that could be helpful to estimate what the person's weightings or imaging is at the same time point. So in this case, Nikhil again designed an architecture using an autoencoder that takes in the T1 weighted MRIs and outputs the T2 weighted MRI of the participant. And he trained this on a large sample that we're hoping to augment in the future. And the validation shows that actually there are small differences in MRI intensity for the imputed modality based on the original T1 weighted MRI. So the idea here is that a person in the future using these approaches could go into the clinic or hospital, and instead of sitting in the scanner for a long time, like half an hour or however long it takes to acquire all these modalities, perhaps in the future one can just acquire one modalities like the T1 weighted MRI shown here, and then using generative AI impute the missing MRI weightings so that the timing required for the scan is much lower, and so the cost saving benefit is obvious in cases like this. So this is a set of preliminary results. One thing about T1 weighted versus T2 weighted MRI in imaging is that their ratio is proportional to the amount of myelin in the brain, which is a substance that is involved in the proper conduction of axon potentials along brain cell fibers in the white matter and gray matter. And myelination is important in a lot of diseases, especially neurodegenerative diseases. There is loss of myelin in Alzheimer's disease, also issues with myelination in other conditions. And so this is a very interesting and nice test to look at how the imputation model works. And so we've been using the NAIRR resource on these projects. And here at the top, you see the true myelin map of the one participant obtained from the genuine T1 and T2 weighted MRIs. And at the bottom, we have the synthesized myelin map. And as you can see, they are very similar. And so we are very hopeful about the potential of these techniques and the use of generative AI to better estimate myelin maps, to impute new modalities in MRI, and to advance the field for clinical care. If any of you are interested in additional information about our work, feel free to take a look at the BBC documentary Mind Over Matter that was presented by Lara Lewington last summer, and our lab's been featured there. Also, we've been featured in the Wall Street Journal and a number of other publications, and feel free to take a look at those. We're very grateful for the ability and opportunity to use the NAIRR computing resources that we've been allocated, and we hope to continue our engagement to train full resolution models. Right now, we're trying to look at smaller resolution, but hopefully we can request some resources for dense GPU nodes or larger resources like the NVIDIA DGX Cloud to train high resolution models. And in closing, I'd like to thank everyone in my lab, our colleagues at USC. This has been the outcome of a collaboration spanning several continents and many teams in Australia, UK, and of course, I'd like to thank our funding from the National Institute on Aging, and I'd be happy to take any questions if there are any. Thank you very much for your time.
Thank you, Dr. Andrei. That's a really insightful, presentation. And if you have any questions, please put down the Q&A section, and it's going to be answered as you ask. Our next guest speaker is Dr. Zhang. He's going to talk about his project title, NAIRR Palette Accelerating Large Foundational Model, Pre-Training with Sparse Communication. Dr. Zhang is an assistant professor in the Department of Electrical and Computer Engineering at Rutgers University. Before joining Rutgers in 2023, he was a computer scientist and lead machine learning group person at Texas Advanced Computing Center. He was a postdoc researcher at AMP Lab, UC Berkeley, and a data science fellow in the Berkeley Institute of Data Science from 14 to 16. Dr. Zhang received his PhD from Department of Computer Science at University of Chicago in 2014, and Dr. Zhang has extensive experience in high-performance computing and big data systems. His research focused on the fusion of HPC and deep learning with a wide range of topics of optimization algorithms, IO architecture, and domain applications. He co-leads the Cyberinfrastructure Trust of the NSF, ICI, CLE AI Institute. He's also the PI of the NSF Diamond and HPC GPT projects. Dr. Zhang received his NSF Career Award in 2024. Thank you, Dr. Zhang, and can you please share your screen?
Yeah, yes, I'm sharing. Okay, cool. Aloha. I'm still in Hawaii. This is my second last day. Okay, so today I'm presenting some work that we did under the NAIRR pilot resources. Okay, so the title is Accelerating Large Foundation Model Pre-training with Sparse Communication. Okay, so it seems like everybody knows that training a large foundation model, especially the pre-training phase, is very expensive, right? If you look at the Llama 7B, this is a small scale one, right? It takes about 82,000 GPU hours on A100 GPUs. But you can think of the 405B from Llama 3.1, that take 16,000 H100 GPUs for almost 2 months, right? And if you look at GPT 8.3 billion model, it takes roughly 25,000 GPU hours per epoch by using V100 GPUs. So we profile the communication overhead using the 2.0 billion model with the global batch size of 512 on neuroimages supercomputer using only data parallelism. And on the right-hand side figure, you can see the communication takes over from 11% all the way to 65% when we scale from 8 A100 GPUs to 128. So communication is dominant. And naively, you can think about, how can we reduce the communication overhead, right? So either we can lower the communication volume. I mean, there are several possible ways. One is to sparsify the gradients. We don't have to communicate, all the gradients or every single step. There's like existing research like deep compression. I can't remember the name, DGC, yes, deep gradient compression from MIT many years ago, right? And for convolutional neural networks, you only need to commute like the top 1% of largest gradients. And the second way is to quantize those gradients, and we can relax some of the precision of those gradients to lower the volume. And the second way we can do is lower the communication frequency or leverage. Third way, which is also to lower the communication frequency by leveraging second order information. And there's like recently people are looking at like muon optimizer to do this. So in this talk, I will focus on gradient sparsification, which was published in MLC's 25, which a few months ago, with my student Min Kai. Good. So if you look at the existing gradient compression techniques, using top case sparsification is kind of like intuitive. You can, for every single iteration, we can simply select the top K gradients with the largest magnitudes, right? But the drawback of this line of method is like the position of those gradients are quite unstructured because they're random, right? And the scalability will be largely constrained by the altogether operation. And the second line of research is to apply low rank approximation, where we use where we decompose the gradient matrix or tensor into two smaller tensors. And in this way, the decomposed matrices are structured, okay, but the overhead is still high. So we did this experiment with 128 A100 GPUs. If we do this power SGD with the rank of 128, you see the computation overhead is 574 milliseconds, which is 12% more than the pure computation time. So so it's even longer than communication time. So we look at the position changes over those over the gradients over time. And what an interesting thing that we see the top 1% gradient distribution is quite stable a long time. So from the 80,000 step all the way down, you see there are clearly several straps of these gradients. That means the largest gradients falls into those buckets. Okay, so that motivates us to design a compression technique that leverages this stability a long time. So we call this algorithm radius. So we perform top K selection for only every T iteration. So we reuse those selected indices in the following T iterations, and this is to amortize the computation overhead. And to select the top K gradient indices after reduce, so all the workers have the same top K indices, and the top K values are structured. Then we use altogether can be the all-gather operation can be replaced by R-reduce. So we can take advantage of this stable structure of the largest gradients. Okay, so we implement the algorithm. For those so for those iterations that we don't require communication, we simply accumulate the errors and we apply this error correction for every t iterations. Okay, and this is a brief breakdown so because we introduced two hyperparameters. One is the density, right, and the other one is the communication interval. So we do a permutation study of these two hyperparameters. And if you look at this figure, so with d equals 0.4, which means we only communicate 40% of the gradients, and for every 200 iterations, we can accelerate the training by 21% for each iteration. Okay, and if you look at the very last column, if we do if we only communicate 10% of the gradients for every 200 iterations, we can accelerate by 36, 37. But we will see the convergence impacts later on. Okay, and this table shows the downstream task performance of the models that trained with the radius and the baseline. So if you look at baseline, right, the first row, so those are the numbers we use with we derived from with Adam W. And with d equals 0.4, t equals 200, right? So you see out of these 5 benchmarks, 5 downstream tasks, we have 3 with higher performance than the baseline. So we say, with radius, with this d equals 0.4 and t equals 200, we have comparable performance to baseline. We're not declaring it's better because there's randomness. So this is comparable. Okay. And we examine the scaling efficiency. You see with 128 A100 GPUs, the scaling efficiency of radius is 42.5%. It's still not very high, but compared to the baseline, it's only 32.8%. Okay, so it improves a little bit, 10%, but it's still considerably low. Okay, so to summarize this work, radius is effective gradient sparsity algorithm and system for accelerating large FM pre-training. It overcomes the limitation of top-case sparsity by leveraging structure and stability of important gradients. And we obtained a 21% speed up on walk-up time, and without sacrificing downstream task performance. And we'll continue working on reducing tree and validation complexity for higher sparsity rates. If you look at our paper, with this density of 10%, we can achieve 36% time reduction without significant loss on downstream task performance. But we were trying to very conservative on the paper, so we only claim 21% speed up. Okay, cool. So during this progress, we noticed that installing software. So we use NERSC promoter, we use Tag Vista and Delta NCSA Delta AI for this study, and we noticed that installing software across machines is really challenging, especially for those novice users or deep learning researchers who doesn't have cyber infrastructure knowledge. As this example shows, I was involved in this open source AlphaFold 2 re-implementation as a project of OpenFold. So we have the data set, we have the neural network, and we have a bunch of machines we want to run. And for every single machine, we need to install software, run on many GPUs, do job management, data management, promise management. It's quite tedious and repeatedly consuming a lot of effort. So we designed this Diamond service that enables users to train neural network across those machines. So Diamond provides a web user interface for users to configure container image using the web interface and launch the job on the machine that they want to run. So this is a simple guided interface for user to build a container, and we have a similar interface for job composition. Okay, and when we are working across machines, they have different schedulers, they have different launchers, right? So we also designed a LLM-enabled tool to port the command line from one machine to another. Okay, and this is being integrated into Diamond. So far we have Diamond, fully tested on Delta AI, Frontera, Lone Star 6, and Vista, Bridges 2, and Anvil. Okay, and we are also supporting ALCF Aurora, tech Stampede 3 because those are Intel GPU machines. And yeah, okay. So yeah, this is my team of Diamond. And if you are interested, please, you can scan the QR code and I'm happy to answer questions. Thank you.
Thank you, Dr. Zhang, for your information. And if anybody has questions, please put on the Q&A section. Our next speaker is Dr. Hari Subramoni. He's going to talk about smart cyberinfrastructure for democratized digital agriculture over the cloud. Dr. Subramoni is an assistant professor in the Department of Computer Science and Engineering at The Ohio State University, USA. His current research interests include high-performance computing and scientific computing, focusing on deep learning, machine learning, digital agriculture, and cloud computing and fault tolerance. He has co-authored over 170 publications in international journals and conferences related to the research areas. With that, I would like to welcome Dr. Subramoni to share his screen, and there we go. Thank you for your time.
Thank you for the kind introduction. I hope my screen is visible. The talk is going to be on, so I'm part of the National Science Foundation funded Icicle AI Institute. We are trying to see how we can democratize high performance computing and artificial intelligence to help various domain users. So in this particular talk, I'm going to talk about how we have been able to do this using NAIRR resources for the domain of agriculture. Now, as all of you know, we face a lot of food security challenges for just give me 1 second. Okay, cool. So we face a lot of challenges in food security. So we are not able to cultivate enough food to feed all of the growing population. Furthermore, nobody is willing to go into farming. So who's going to train the next generation of agricultural workforce? Finally, farmers are not able to use the modern evolution in high performance computing or AI. How can we enable farmers to take advantage of these advancements in computing and technology to their field so that they are able to do farming much better? So long story short, this is what a farmer does for most of the time. They look at a soybean field or a cotton field or a broccoli field and say, is this healthy or is this not healthy? Now, me personally, I'm a computer scientist. I have no clue. But a farmer would be able to look at a particular leaf and say that, hey, I have nutrient deficiency here. I have crop blight here. I have, let's say, infestation here, all of these things. So can we use AI to make these predictions and find answers to questions like, can you tell me which portions of the field are defoliated? So that farmers can make smart choices. So right now, farmers might spray their entire field with herbicide, fungicide, or weedicide, which is going to waste money as well as affect the environment negatively. Can we say that, hey, in your 10-acre farm, only this particular portion, maybe 600 by 200 square feet portion is affected by, let's say, blight, and that is the only portion that you have to spray. How can we do this in an easy to use way, taking advantage of computing and AI? That is what we are trying to do. We are trying to analyze various crop health issues using high performance computing and artificial intelligence enabled cyber infrastructure so that farmers can make crop management interventions on the field. So we span the entire computing continuum from on-field sensors, near and far edge devices to cloud solutions like NAIRR and HPC systems and data centers. So we work with a lot of partnerships, farmers, co-ops to identify crop health issues. For instance, how much residue cover is left on the soil, what is the aggregate size, what development stage the crop is in, and analyze that using computing and AI techniques to make crop management interventions. So should I apply more nitrogen? Should I water the farm more? Should I apply fungicide, herbicide, or weedicide? So we try to create cyber infrastructure which can collect, process, train, and infer using data on the field and allow farmers an interactive way to make on-field decisions. Now, what is the big deal here? So there are several ag tech companies who have proposed similar solutions. So for instance, John Deere has a product called See and Spray. So it is able to detect green weeds on a background of green foliage. Even just this very simple solution for soybeans has been able to reduce herbicide application by 70 to 90%. Just imagine one solution. However, very little work has been done to expand beyond weed control in major crops, as well as nothing has been done to figure out how we can reduce the application of fungicide, insecticide, or how can we like control the application of nitrogen. So nitrogen is a $100 billion industry. So if you are able to save 1%, that's $1 billion every year. Unfortunately, there is a lack of software infrastructure to uh, correlate and collate all of these things to generate final solutions for end users. So we try to create this software solution because large companies lack the bandwidth or the incentive or the technical ability to tailor their solutions to the needs of individual farmers. Okay, so currently there are only green on green technologies. How will we make this applicable to other crops like cotton, corn, broccoli, cherry, strawberry, so on and so forth? So this is where we come in. We aim to democratize agricultural workflows through low-cost public access hardware and simple composable interfaces that can adapt quickly to the needs of practitioners. And that's where solutions like NAIRR, sponsored by federal agencies, come in. So broadly, what we are trying to do is, can we collect data on the field, transfer it through wired or wireless mechanisms to either an edge device or HPC and cloud centers like NAIRR, and generate prescriptive answers that can be fed to smart tractors to make precision application on field. So this is an overview of the overall pipeline that we have created. And in this pipeline, we have multiple components which have all been deployed on NAIRR resources or are interacting with NAIRR resources like AWS or Azure for authentication and data movement, inference and training, and final visualization of results. So all of these have been deployed on NAIRR resources. And these are all of these are downloadable from our website. You can we have containers available which you can deploy on AWS or Azure and try all of these things out. Now, some nitty-gritty details as to what storage and compute requirements we need. So typically, we collect gigabytes, multiple gigabytes, terabytes of data and process them. So all of these have to be stored, and the NAIRR storage on AWS and Azure really helps us for this. And our computing requirements are also pretty high, and we use a lot of 800 and other GPU computing and CPU computing resources available on these high performance clouds to accelerate our compute operation. So let me show a demo of our entire pipeline working on the HPC and cloud instances.
Welcome to the future of agriculture. This is Icicle's AI cyber infrastructure for end-to-end processing of digital agriculture use cases. As a user of this framework, you can easily compose and manage your jobs all from one convenient and user-friendly interface, which is connected to an HPC backend where your jobs will be accelerated on modern GPUs and CPUs in a distributed fashion. Let's get started. The dashboard is your control center. Here you can select from many modules to compose your new jobs. Depending on the nature of your job, you can take advantage of one or a combination of modules, including data collection and pre-processing, image labeling and high-performance model training, model inference, and visualization of results. The dashboard provides a comprehensive overview of your resource usage, data storage, and processing times. Let's walk through an example of creating a job which uses all of these different modules. The use case here is classification of growth stages in a cornfield, where the goal is to train and use a deep learning model to analyze phenotypic data and provide detailed insights on the growth stage. Let's create the new job. The first stage is data collection and pre-processing, where you can import your data from Model Commons or upload it from your edge device directly. Once the pre-processing is done, we will have access to download and examine the pre-processed dataset. For now, let's move on to the next step, which is the model training. This page shows a list of model training options. The values will be populated by default for beginner users. At the same time, advanced users can adjust the training hyperparameters. The domain expert will then provide a number of label images for each class. This current page shows the first growth stage class, as shown in the very top. We can navigate through the samples and select representative images. Then we'll click Submit to move on to the next class to be labeled. Once the images are labeled for all the classes, distributed DNN training will be submitted automatically on behalf of the user. For inference, the user can select the inference task they wish to perform. In here, our task, again, is growth stage classification. Then we can select the model to use for inference. We can hit Submit, and that will take us back to the job overview page. Now, let's go to the final stage, which is visualization. The heat map is a view of the entire field. There are several blocks, each one of them is color-coded based on its predicted growth stage. The user can click on individual blocks, review the zoomed-in images, and provide feedback to the system if they think the classification is incorrect. Finally, let's look at the results gallery. Here, the user can examine individual images, their predictions, and the confidence score provided by the deep learning model. This job is now considered complete, so let's quickly go back to the dashboard. We can see the updated account statistics with the remaining SUs and storage. The new job list now shows the job that was just completed, and the user always has the option to go back and visit completed jobs. This concludes our overview of the framework, so thank you very much for listening.
I hope the audio came through this time.
Welcome.
So this is what we have been able to create, and this is not work that just our group has done. This is a collaborative work between multiple institutions across the country who are all participating in the NSF Icicle AI Institute. So kudos to all of them for enabling this. So that with that, I come to the end of my talk, and if there are any questions, I'm more than happy to take them. Thank you.
Thank you, Dr. Hari Subramoni. We have two questions for you, and we have time also. So the first question is, without turning the conversation into deep architecture discussion, what cloud services in AWS and Azure are hosting the compute and storage demands?
So on AWS, it is the EC2 instances. On Azure, I need to go and check. I don't remember it off the top of my head. We use a parallel cluster feature on AWS so that we are able to have some services running. So let's say when a user requests a new job or submits a new job, there is something there to accept the request and start off the training or inference processing. If you can send me an email, Chase, I can get back to you with more details on the exact instances and the other details.
This
Sorry,
next
go ahead.
question from Jose. It says, "What's your website URL? Link is missing from your bio at Ohio State."
Okay, so I can give that.
We'll
So
type the answer right. We'll type the answer right now. And the next one is ICICLE is available, so we can have account and use it.
yes, Anas, it is available. Please send me an email. We can set up access for you. We are trying to expand beyond the initial agriculture phase and trying to see if other users are able to take advantage of it from the domains of geosciences, animal ecology, political sciences. So when we looked at it, we found out that the requirements for various users are kind of similar. How do I gather data? How do I process data? How do I train models? How do I infer with those models? But the problem seems to be that even though bits and pieces have been done, no work has been done to put them all together into pipelines, end-to-end workflows that can be usable by domain scientists. So we are trying to reach out to more people. and enable people to use that. And NAIRR has been significantly helpful for us in that regard by giving us the necessary compute resources, so that compute and storage resources, so that we can, let's say, engage end users and other stakeholders to utilize what we have developed as part of the NSF-funded effort.
We can take one more question from Vardhan. It says, is dashboard available for public use yet?
Yes, we are. So if you saw, a bunch of these have been released. We are actively working with various stakeholders. Send me an email. I can get you access to the system. Thank you.
Yeah. Thank you, Dr. Subramoni. We're going to move on to our next panelist who is here, Dr. Avinash Sahu. He's going to talk about his project, Advancing Colorectal Cancer Diagnosis, a multimodal AI Copilot for real-time endoscopy. So in 2023, Dr. Avinash Sahu was recruited from Dana-Farber Cancer Institute and Colorado University as an assistant professor in the University of New Mexico Comprehensive Cancer Center. He holds a PhD in computer science from University of Maryland and National Institute of Human Genome Research Institute. He completed his postdoctoral training at Harvard and Dana-Farber Cancer Institute. Dr. Sahu leads the Tumor AI Lab, focusing on the nexus of machine learning and precision oncology. His distinction includes Michelson Prize from Human Immunome Project and recognition from the Irving Foundation as the top 40 young cancer immunology scientist. His innovation in the precision oncology earned Invention of the Year in 2016 by the University of Maryland. Additionally, in 2020, he received the K99R00 award. It is now advancing an AI-based precision oncology predictor. Thank you, Dr. Sahu, for joining us and for your presentation.
Thank you. I hope you can see my right screen. Can you see my screen?
Yes.
Yeah, okay, fantastic. So yeah, today I'm going to talk a little bit a more broader thing, which sort of also cover the project that is supported from NAIRR. And this is, we are developing large language model to interpret different biological data and signals. So, you know, there are really the trend of people who are taking genetic screen, and rightly so, is increasing year after year because genomic sequencing is becoming cheaper, and you know, more and more people are getting recommended for genetic screen. And in many cancer, genetic screen and a follow-up with a preventive surgery is only an option. For example, in ovarian cancer, where you know the efficacy is almost 96%. But what happens in most cases of my this video is that when most half of the women, when they receive half of women who are at the risk receive their genetic screen, they get this sort of ambiguous results, and the reason is there are problem in our current ability to interpret genetic sequence. So for example, this T53 gene, this is like most highly mutated gene in cancer. So there are three different categories for it. You know, there is a category that different mutation in the same gene could be benign, it could be pathogenic. So, you know, there could be a specific recommendation where if some patient have mutation or a person at a risk have mutation, then they can take preventive surgery. But half of the patient get this sort of in their genetic screen annotation that we don't know right now what is the significance of this mutation is, what is the significance of the variant, right? And it appears in really this super important key gene in the cancer, and it kind of creates this kind of a diagnostic limbo for the patient, right? And more generally, if you talk about what is our ability to understand any protein sequence, so there are almost 222 million protein sequence, and only out of that 0.2% of them are characterized. So 99.8% of protein sequence, we don't know what they do, right? And so that's really a big challenge here. And generally, only experimental method to characterize them is definitely going to be insufficient, right? So what we are talking about is, can we use computational model, or like, for example, can we use ChatGPT to understand it, right? Like, where you give a protein sequence and ask question like, what is the function of this protein? What is the function of this mutation? And the problem is, current ChatGPT is not trained on that. Like, it cannot interpret protein sequence. It can recognize that this is a protein sequence, but it doesn't understand it. In its vocabulary, literally, protein sequence is not there, right? So a straightforward answer to this problem could be that we can increase the vocabulary of current large language model, right? It have, in its current vocabulary, there are all this English language. Can we add protein sequence? So each of this represent a specific amino acid sequence that constitute all of the protein in the world, right? So we can just increase this vocabulary. The problem is that the grammar or semantics between protein sequence and English language is incompatible. Protein sequence follow this very different biochemical rules, unlike what natural language brings in. It depends on 3D structure, which there is no concept of 3D structure in English language or any other language, right? So there is an inherent incompatibility on this. So to address this issue, Ala Jararwe, who was a PhD student, he graduated this year and he's starting his faculty position in October, I guess, this year. He came up with a really simple idea. He said that, let's not directly increase the vocabulary. Can we sort of learn an adapter which sort of takes a protein sequence and then sort of transform this protein sequence into a space, a latent space, which is similar to the text space. So can we sort of translate the protein space to a language space? And so we come up with a simple framework. We have a protein sequence. We can encode them into some embedding space. And then the main thing is to learn this projector, which will sort of takes the sequence space and then convert into a text space. And then if it is in the text space, then you can sort of process with any language model, right? So now we don't have to sort of train this new language model. We can, if this is very similar to the text space, we can take existing language model. We may need to fine tune it a bit, but we don't have to train it from scratch. And that was really a key here, right? And then that can sort of help us to interpret the protein sequence, right? Now, the question is, where do we get the training data? We need, protein sequence, and then also this sort of a text describing this protein. And if there is a related question and answer, right, you would need protein sequence, question and answer. The way Ala did this, created this database is that, people have worked for almost a century, more than a century, on sort of understanding different protein sequence, different genomic sequence. So and they're all published, right? So, and most of them are publicly available. So we process 24 million PubMed abstract, and this is where we use NAIRR for. So we start with the PubMed article. We use one of those language models, and then sort of prompt it to sort of generate question and answer, which could be answered from the sequence, right? And then we have this, we need some sort of, what do you call this? We need to make sure that there is no hallucination and there is, the names are correctly mapped and things like that. So this part is basic bioinformatic things. But the main thing was, this was more sort of computational intensive, where we use NAIRR. And so we created almost, right now we trained the model on 47 million protein question and answer data point, and it looks something like this. You have a sequence, you have a question and answer, you have a question and an answer. We input to the model the protein sequence and then question, and then train the model to give this specific answer, right? And we have almost 47 million data points like this, right? And then we showed that you can easily sort of beat general purpose language model. It's not surprising because they were not meant for this, but we can also sort of outperform many of the models which have been sort of trying to do something similar, right? And this is what we use. NAIRR resources has been super helpful. In addition to the protein domain, we are also working using a very similar strategy to analyze endoscopy image. These are colonoscopy images. We use a very similar idea where we encode it into this image space, but then we use a projector to project that into the text space and then process the language model. So I think that's all I have. One of the things which we are really looking forward is when it comes to, we have trained protein to text on interpreting proteins. So you have to give the entire protein. When it comes to the variant, each protein could have thousands of variants. And so we are talking about order of magnitude, higher number of data points and training, and that's where we are we are looking forward whether we can get higher resources. We have access to Oak Ridge compute resource, but they are AMD based, and a lot of things are not supported in AMD GPU. So NAIRR has been really helpful for us. And so a lot of people contributed to this work, and I'm happy to answer any question.
Thank you, Avinash. If anybody has any questions, you can just put in the Q&A section. I don't see any questions for you now. No. So I think with that, I think we should conclude our seminar. Thank you all for joining us. Thank you, Dr. Sahu, Dr. Andrei, Dr. Subramoni, and Dr. Zhang for joining and giving a wonderful presentation about their work. And thank you, NAIRR Pilot, for supporting such a brilliant research. And I would like to also thank the NAIRR User Experience Working Group for helping coordinate and organize this panel. And with that, I will conclude the panel. Thank you all for joining. And
Thank you for having us.
Please email the speakers. Thank you.
Bye.
Thank you all.
Thank you.